Welcome everyone, my name is Michał and I'm Ensembl Outreach Officer. Today we'll be talking about genetic variation in Ensembl. There are different types of genetic variation and I'd like to start with introducing a variable number of tandem repeats. These are basically short repeats of few to several nucleotide units, repeated one after another, and we can distinguish two types of them. Microsatellites, when the nucleotide units are a bit shorter, and mini-satellites when the units are a bit longer. And microsatellites are also known as STRs. There are also short variants, which are defined as genetic variants smaller than 50 nucleotides. Here we can distinguish single nucleotide polymorphisms, so substitutions of a single base, and also small insertions and deletions. Finally, there are also larger structural genetic variants. Those can be larger than 50 base pairs, but in fact they might affect larger chromosomal chunks and also entire chromosomes. And here we can distinguish copy number variation, including gain and losses, but also inversions when a chromosomal region is inverted at the same position, and translocations, which are basically genetic regions moved to a new position, either within the same chromosome or different. We don't have genetic variation data for every single species in Ensembl, but we have it for a good representation of them. We also have genetic variation for some non-vertebrates in Ensembl genomes. Now I'd like to talk about the Ensembl variation process. Where do we get our data from? How do we process it? How do we curate it? So we first import both somatic and germline variation data from publicly available databases. There's quite a few of them, as you can see in the slide. I'm just going to mention a few of them. So we get lots of our germline variants from dbSNP. We also import structural variation from DGVA and human somatic variation in cancer from Cosmic. And we get most of our non-human variation from EVA. Here you can see some of the sources of genetic variation in human. You can see it's quite heterogeneous. We import both germline and somatic variation from different resources. You can find a list of all the resources under the link below. And you can see, for example, we import from dbSNP alone over 600 million variants for human. As I mentioned before, Cosmic is a curated database of somatic mutations in cancer in human. And one thing to remember is that Cosmic data is not entirely public. But what I mean, you do need to have your own license if you want to fully access their data. And those licenses are free for researchers and academics. However, we don't know if people coming to Ensembl have their own licenses. Therefore, we are not allowed to show you the allele information or allele frequency. We only show you the position of those important variants, and we also show you the consequence predictions for all possible alleles based on the variant location alone. And then we link out to Cosmic for further exploration. We don't import variants blindly. We do perform our rigorous QC, so we map those variants to the reference assembly. We then check whether at least one of the alleles matches the reference. We also check for IUPAC ambiguity codes. If you're not familiar with IUPAC, it stands for International Union for Pure and Applied Chemistry. They're responsible for the nucleotide codes. So apart from ATCG, you might also see different letters in the genetic code. For example, R, which would stand for either A or G, so purine. We also exclude suspect variants if there's something fishy about them. So for example, if they map to multiple regions. We also add value to that data. So we look for further information. We mine different resources, databases and publications to bring as much information as possible. We provide phenotype and disease information, allele frequencies from different population genetic studies, and list of citations. Here we have some of the population genetic studies that we calculate allele frequency from. One of the biggest ones are 1000 genomes. which is low coverage whole genome sequencing data for around 2,500 individuals from 5 major continental populations, and these are all healthy individuals. We also have Nomad, which is a mixture of whole genome sequencing and exome sequencing. It's not as diverse in 1,000 genomes. It's biased towards the mostly overstudied populations like Europeans. However, the advantage here is a really large number of individuals, which is above 100,000. And those individuals are a mixture of both healthy and diseased individuals. And finally, we do have some disease cohorts like UK10K and TopMed. Finally, we do not only import the variation data, we also perform some analysis ourselves. So we provide the variant consequences based on where in the genome and in the gene those variants fall. We provide protein functional predictions, linkage disequilibrium data, and conservation across species. So the variant consequences are described using sequence ontology nomenclature, which is a standard language describing sequence-based features. So variant consequence is classified based on where in the gene or genome the variant falls. So you can see here, for example, we have a variant falling in promoter. It's going to be classified as a regulatory region variant. But there are some variants which might have multiple consequences, like for example, this variant over here, which falls both in the promoter but also in 3' UTR. And you can see all those consequences are color-coded. Here's a list of all possible consequences in Ensembl. You can see there's quite a few of them. The color coding is consistent throughout the website. I just want to point out that they're listed here from more severe towards less severe. And you can see the more severe consequences have those danger colors like reds and yellows and oranges. And then the less severe consequences like synonymous variants or intronic variants have these bluish greenish colors. And finally, we also have some regulatory region variants in brown. We also provide pathogenicity predictions, which are various algorithms scoring, assessing the effect of amino acid substitutions on the protein. And they take into account different properties, like for example, chemical similarities of the amino acid. So if a missense mutation changes amino acids from polar to non-polar, that might affect the protein more severely. Some others might take into account 3D protein structure and domains, or conservation and sequence homology. So for example, if a variant falls in a conserved region, is more likely to be deleterious. It's important to remember they provide predictions, not facts, and most of them haven't been functionally or experimentally validated. So they should be taken with a pinch of salt, as they might sometimes disagree. Here we have a list of some of the pathogenicity predictors that we provide in Ensembl. So I would say the first three are more commonly used. We've got SIFT, polyphen 2, CAD, and some others. What I wanted to point out that they're all a bit different. You can see some of them range from 0 to 1, while others from 0 to 100. They all use different cutoffs for pathogenicity classes. They use different terminology. In some cases, low values might be deleterious, while on the others, it's the opposite. I just wanted to stress that SIFT is quite popular, as it's available for many different vertebrates, not only human, while CAD is a little bit different, as CAD scores are genome-wide. They're not only available for missense variants like the others, but they're also available for non-coding variants, including SNPs and indels. I want to talk about the reference alleles now. As you might already know from the first webinar, when Ben was talking about the human genome assembly, human reference genome is composed of different people's DNA. That means different chunks of the reference might come from different people. However, every single chunk is real person sequence, and they're not consensus. So for example, if this T allele over here indicated in orange is found in the reference, that means it just happened to be in that person's DNA. So that CM individual carried the T. If there is an alternative G allele in this position, an ensemble always gives the reference allele first. What is important is that the fact that the T is found in the reference in that individual does not imply its allele frequency, evolutionary relationship, or disease association. So this reference allele can be either major or minor, ancestral or derived, and by derived I mean evolutionary new, or it can be either healthy or disease associated. Furthermore, as you can see here in this cartoon, genetic variants affect both strands, so they're double-stranded because of the complementary nature of DNA. As you can see, the alleles of this genetic variant can be either reported as T, G, or AC, depending on which strand you're looking at. So how do we report them? So there's a common consensus, there's convention accepted by most of the biggest databases that alleles in the database are generally reported on the forward strand, regardless of whether the gene is on the forward or reverse strand. However, if you're looking at the sequence of reverse-stranded gene, like in that example, we're going to show you the alleles as they are in the gene or the exon AC. The literature is a bit of a minefield as people report the alleles on either strand as they want, and often they don't even indicate which strand that they were reported on. So bear that in mind. Finally, you can get our variation data from our Ensembl main page at www.ensembl.org. You can mine this data using our Variant Effect Predictor that I'm going to be talking about later on. You can also mine it using our online export tool, which is Biomart, which I'm going to be talking about during our last webinar in the series. And finally, you can also access it programmatically using our APIs. So now we're going to have a short demonstration. I'm going to look at the AGT gene and explore a variation in that gene. So let me jump out of my presentation and go to the Ensembl Genome browser. You can go to the Ensembl homepage by typing www.ensembl.org, and now I'm going to look for our gene of interest by typing AGT in the main search box and hitting enter. So the variation data can be found across many different tabs in Ensembl, and we're going to explore a couple of them, starting with the gene tab. So I'm going to click on human gene, which is the first hit. And now using the left-hand side menu of available views, I'm going to click on sequence. You can visualize variation data in basically any sequence view in Ensembl. To do so, just click on configure this page and choose show variants. Yes, I'm going to choose to show them with links. We also have some filtering options over here. You can, for example, filter variant by consequence type or by source of variation. I'm not going to filter for now. I'm just going to click save and close. It might take a little bit to load, depending on how much variation data there is. Here we are. So you can see all our variants are highlighted here in different colors. You can find a legend here on top indicating what type of variation those are. And I'm just going to hover over this R, which you can see stands for A or G. If I click on this variant, it's going to open pop-up menu with the variant's information and the variant name as interactive link, which you can hit. Here you have the source of the variant, also the genomic location, and also alleles here given as TC. So if I hover over it again, you can see they're here indicated as A or G. Why the mismatch? It's because the variant alleles here reported independently of gene are always given on the forward strand, as mentioned before. This gene is a reverse-stranded gene, which is indicated here in the header by -1, which stands for reverse strand. Therefore, the alleles in the sequence here are shown as they are in the gene. can also explore variation within a gene by going to the variant table. It summarizes all the variants found within a gene. So you can see here we have multiple columns. We have a variant ID, which is also an interactive link. We've got genomic position, alleles, and many other information, including source and evidence. The more evidence, the better. That means the variants are more likely to be real. Then we have the consequence type and some other information. You can also manipulate and filter this table. I'm going to click on consequences, then turn everything off and select a missense variant, then apply. Now you can see we've got extra information. You can see these are all a missense variant. You can see the amino acid. You can see the position of the amino acid within the protein, and you can see some pathogenicity predictions. They do not always agree, as I mentioned before. Here you have some of them say that this variant is totally fine, while others say that it's likely deleterious. If you're colorblind and you can't see the colors, just hover over and you're going to see the phrasing as well. To remove the filter, simply click on the X. Now I'm going to show you the structure variants. Let's click on the structure variants view in the left-hand side menu. So you can see the structure variants are given here in 2 tracks. We have one track for small structural variants, which are shown individually, and then we have one track for larger structural variants, which are condensed into one bar found across multiple studies. We have a legend over here, and below you can find a table of all structural variants falling within that gene, again with their IDs as interactive links and coordinates. Let's explore the phenotypes now. Here you can find phenotypes associated both with the gene, like for example hypertension, but also phenotypes in diseases associated with variants falling in this gene, listed here in this table. Furthermore, you can also find phenotypes associated with the orthologs of this gene, which might be useful if you're working with a gene that, for example, is not very well studied and not very well understood in humans. But, for example, there is available a knockout of a mouse ortholog, which might be a functionally equivalent. But also the other way around, if you're working with an organism that is not very well studied, you might have some information here from human orthologs. Let's now explore the variant data in the transcript tab. I'm going to click here on the transcript tab just next to the gene tab. And there's one particular view only available in the transcript tab that I wanted to show you now. Let's click on haplotypes in the left-hand side menu. This table here lists all protein haplotypes found in 1000 genome populations and their frequencies. And by haplotypes, I mean group of variants found together in the population, so inherited together and segregating together in the population. And you can see, for example, the reference haplotype is actually not the most common one. There is another haplotype different from the reference by one substitution, which is more frequent. I'm going to click on the third haplotype, which differs from the reference by 2 substitutions. And that takes me to more detailed breakdown of the haplotype frequencies in different 1000 genome populations. And if you don't know what any of those acronyms mean, just hover over to see a description. If you scroll down, you can find alignment of different haplotypes. You can see both the protein and coding sequence alignment. And you can see here you have the 2 substitutions, which differ my haplotype of interest and the reference haplotype. And if you go further down, you can find the protein sequence, but also a table listing all 1000 genomes, individuals and samples carrying a given haplotype. So if you wanted to follow up on certain haplotype or variant functionally, you can order a cell line from Coriell carrying that specific haplotype. Let's now move to the location tab. I just wanted to show you where you can find variant information in the region in detail view. So as you know, by default, we show you a limited number of tracks. To add more tracks to your view, go to configure this page where you can find variation section. I'm going to click here, which takes me to a list of all available variant tracks. You can see some of the tracks are already turned on. Here we have a section of tracks grouped by population from 1000 genomes. Here we have some phenotype associated variants grouped by source and so on and so forth. And here finally on the bottom you can find structural variants as well. Let's say I'm interested in short common variants found in East Asian populations. I'm going to turn the track on as expanded with name. Save and close. You can see now that our tracks have been added to the view, and here we have the common variants found in East Asian populations. I can click on them individually. The variants are represented by those lines. Clicking on the variant should open the pop-up menu with variant information, RS number, so variant ID from dbSNP, variant position, and so on and so forth. You can see that this variant is intronic variant, which is indicated by this dark blue color. And actually, if you scroll up, you see that this variant falls in the intron of the AGT gene, which makes perfect sense. Okay, let's explore the variant tab. So Ensembl has a special tab dedicated to a variant itself. I can search for variants using the search box here in the corner. I'm going to type RS699 and hit enter. Let's click on the human variant, which is the first hit on top. That takes me to the variant tab summary view. Here you can find the most severe consequence of this variant, which is missense. You can find allele information with the reference allele given as first. And then you also have ancestral allele information and minor allele frequency. You also have CAD and conservation scores. You've got location of this variant and other known variants in the same position. Here you have different evidences. So you can see this variant's been reported in many different studies, which is good. That means it's more likely to be real. Finally, we have different available views displayed here as different icons. They correspond to the left-hand side list of possible displays. Let's click on genes and regulation now. This table here summarizes variant consequences. You can see that the specific genetic variant only hits one transcript of one gene. It's a missense variant in this gene. You can see its position in the transcript, in the coding sequence, in the protein. You can see the amino acid substitution from M to T, and also a position in the codons. And you can see also that this variant is predicted to be benign by all different pathogenicity predictors. And on the bottom, you can also see EQTL information, so either increase or decrease of a gene expression which correlates with this variant. You have a strength of the association, the effect size, and also a tissue where the gene is expressed. This variant doesn't fall in any regulatory regions, but if it did, it would be listed here. Let's now go to the population view. I'm going to click on population genetics. Here we have information on allele frequency across different populations. Those pie charts represent the different allele frequencies across 1000 genomes populations. If you don't know what those acronyms mean, again, just hover over, you're going to see the label. And we can see there's a little bit of population differentiation going on here. So for example, in the European populations, the major allele is A, while for example, in the African populations, A is less frequent, is only about 10%. You can also see more detailed breakdown of allele frequencies per subpopulations. To do so, just click on the plus to expand. Now we can see extra pie charts showing the allele frequency across different subpopulations within a continental group. Here on the bottom you have more detailed breakdown of allele frequencies and genotype frequencies across different population genetic studies, including 1000 genomes, but also NOMAD, TOPMed, and UK10K. So as I said, apart from allele frequencies per population, you can also see the genotype frequency. So frequency of heterozygotes and homozygotes in given populations. And the last thing that I wanted to show you in the Variant tab is the phenotype data. Let's click on phenotype in the left-hand side menu. Here we have all phenotypes associated with this specific genetic variant. So here we have phenotypes and diseases names. We have a source from where this association has been imported. We also have evidence, which you can expand, and that will show you a list of citations. Finally, we also have a clinical significance, and you can see this variant is a risk factor for hypertension. You have the associated allele, and also some statistics. So you've got the strength of this association. You can also click on the phenotype that you might be interested in. I'm going to click on hypertension, which takes me to a phenotype tab. This tab lists all genes and variants associated with the given phenotype. You can also filter this table to search for either variants or genes or structural variants or even EQTLs. So that's all I wanted to show you in the transcript tab. Let's now go back to my presentation to talk about VEP, so the Variant Effect Predictor. VEP is a tool that allows a variant annotation, and it doesn't matter whether the variants are known or whether they are novel. So let's say if you got results from your sequencing experiment and you found some variants, VEP can annotate them for you. What it does, it takes a list of input variants described in different formats, and then it talks extensively to Ensembl, pulling out all sorts of information you might be interested in. This is the list of formats that VEP can take in, starting with the default Ensembl format, which is a tabular format with the first column standing for chromosome, second column start position, end position, reference allele, alternative allele in the fourth column, and finally the strand. The forward strand indicated by 1, the reverse strand indicated by -1. The strand column is optional, you don't have to provide it. However, if it's not given, VEP is going to assume that the strand is forward. We then have a standard VCF format. You can also input various variant IDs or HGVS notation, which basically describes variant position within a feature, so within a gene or a transcript. And finally, you can also use the new dbSNP format, SPDY. Here we have a list of features that VEP can output. You can query different gene sets, including our Ensembl GenCode gene set or NCBI's RefSeq or even merge databases. You can then find out whether your variant falls in regulatory regions, also whether it's known or novel. You can get allele frequency from different population genetic studies. You can also get pathogenicity predictors. We provide a bunch of them. And finally, you can also get phenotype and disease association from different resources. There are many VEP plugins. Those are little extensions that add extra functionality to VEP. They can filter or manipulate the output. For example, there's a plugin called Draw. We can draw your variant within a transcript. They may use external data or code. Some of them are available on the web, while all of them are available with the script. And there are many different plugins for different pathogenicity predictors. There are different ways of using VEP. You can use our online version of VEP, which we're going to do in a minute in our demonstration. You can also use the script version. It doesn't require much scripting. You just have to run it. And finally, you can also use VEP using our API. VEP is widely used in both a clinical environment and in the industry. It's also used by many international consortia. Here's the list of species with variation data in Ensembl. However, it doesn't matter if we have variation data for a given species or not. You can run VEP on every single species in Ensembl and everything in Ensembl genomes. Furthermore, you can run VEP for species and genomes that we don't have in Ensembl. So if you happen to work with a unicorn genome, for example, you can also run VEP on it. To do so, you have to set up your own cache. Cache is basically offline VEP. It's a bunch of files and data that web needs to do its job so that it doesn't have to communicate with our database. The advantage of using cache is that it speeds up your analysis. You can keep your data private so that you don't have to submit your data online. It's good, if you're working with sensitive patients' data. You can use pre-built caches for our Ensembl species, and you can also make your own cache if you have GTF and FASTA files. Here's our web publication. If you use web, please cite it. It's best to see it in action. Now we're going to have our second demonstration. So we're going to look at a set of 4 variants, and we're going to see where in the genome they fall, which genes they affect, what are the variants' consequences, and so on and so forth. The variants are described here using the standard default Ensembl format. The dash here indicates deletion. Strand is given as forward, indicated here by this plus symbol. And there's an extra column containing information on a variant name, and the variant names were made up for the purpose of this exercise. So we've got variant 1, 2, 3, and 4. I'm just going to jump out of my presentation now. I'm going to copy our input data, and I'm going to go to the Ensembl genome browser. I'm going to hit web here in the blue bar on top. You can find web documentation and help in this page, but also the script version of it. I'm going to just click on Launch a Web using the web interface, which takes you to the data submission form. Human is set as default. First thing to do is to give our job a name. I'm going to call it Demo. And now we are ready to input our data. If you forgot the specification on different data formats, you can always click on the examples, which will show you the correct formatting options. I have my data copied, so I'm just going to paste it here. Alternatively, you could upload your data as a file using this browse button over here to find a file on your machine. Or you can also provide a link to a file hosted online, for example, on an FTP site. We then have to choose the transcript database we want to compare our variants against. By default, the Ensembl GenCode is pre-selected for you, but you can also choose GenCode Basic and NCBI's RefSeq or merge databases. We then have a bunch of additional configurations. We're going to explore them one by one, starting with identifiers. So here you can see a list of identifiers that web is going to pull out for you. We're going to get gene symbols and transcript versions, but you also have additional options here. If you don't know what any of those mean, just hover over and you're going to get a description. So let's say I'm also interested in HGVS notation. I'm going to select that. We then have variance and frequency data. I'm going to expand this. So first of all, web can tell you if your variants are known or whether they're novel. By default, web pulls out all collocated variants, so all variants known for those coordinates. And you can also pull extra information which relates to those variants, like for example, allele frequency in 1000 genomes or nomad. I'm going to select nomad as well. Furthermore, you can get a list of citations for those collocated variants. We then have additional annotations. So you can see we get transcript biotype, we get different transcript scoring algorithms that you might remember for our second webinar, like for example, transcript support level, APRIS and main. You can then choose to find out whether your variant falls in protein domains, which I'm going to select, or whether your variants fall in any regulatory regions. And finally, you can also get information on related phenotypes, which I'm going to select. You can also get a bunch of different pathogenicity predictions. You can see some of them are pre-selected. I'm going to also select CAD. Additionally, you might also be interested in whether your variants are predicted to affect splicing, or you might also get some conservation information and ancestral allele. Finally, there's an option to filter the variants. Beforehand, you can filter based on consequence and allele frequency. However, it's always better to first run the full query and then filter your data afterwards. Once you're done choosing your options, then hit run. Here we have a job table listing all your web jobs. You can see our job is queued. The page refreshes every 10 seconds, so you don't have to refresh it yourself. If you don't want those jobs to get lost with time, you can save them to your own Ensembl account, and Ensembl accounts are totally free. You just have to register here on top of the page. You can also edit and resubmit the job. You can share it with your collaborators, or you can delete it. Now we can see the job is done, which is indicated by this green label. All you have to do is simply click on View Results. Here we have a graphical representation of our results. You can see we processed 4 variants. One of them is novel, they fall into 2 genes, and 7 transcripts. One of them falls in the regulatory region. You can also see some graphical summary here in this pie chart. Let's have a look at the results table on the bottom. So as you know, we processed 4 variants, but when you have a look at this table, you can see it's quite big actually. It's because one variant might have multiple different consequences across different transcripts of different genes. So you can see here we have 1 row per consequence. For example, this variant 2 is falling in different genes, but also regulatory region, but also multiple transcripts of those genes. So you see it falls in 4 different transcripts of the COQ4 gene. Let's have a look at the columns. As I've just mentioned, the first column is the variant name that we made up, we provided in our input. Then we have the genomic location and allele, followed by the consequence. We have a gene, symbol, and ID, but also transcript IDs. We wanted web to print AGVS notation as well, and here we have AGVS-C, so position of a variant within cDNA, and then we have AGVS-P, so position within a protein. We also have cDNA position. You can see that this coding sequence position agrees with AGVS notation, and then protein sequence position also agrees with HGVSP notation. If a variant is a coding variant, you can see the amino acid. There's only 1 amino acid as this variant is synonymous. Here we have amino acid substitution for missense variants. You can also see the codon position. Finally, if we are dealing with an existing variant, you'll see its known identifier here in this column. You can see all of those are known. If I go to the bottom, you can see if the identifier is missing, that means those variants are novel, haven't been identified before. We then have some transcript scoring algorithms. You can see we've got main, we've got transcript support level, APRIS, and also some pathogenicity predictions for missense variants like SIFT and polyphen. If a variant falls in known domain, it's going to be listed here in this protein domain section. We then have allele frequency information starting with 1000 genome allele frequency, followed by nomad allele frequency in specific populations. We then have clinical significance and motif name if variant falls in a transcription factor binding motif. Like in case of those regulatory variants, you'll see the motif ID. Finally, we'll also have CAT scores and associated phenotypes in case of known variants. Okay, great. You can also manipulate and filter this table. So for example, if I wanted to filter based on consequence, you first have to choose the column that you want to filter on. I'm going to go for consequence. You then have to choose the logical operator. I'm going to leave as it is. So I'm going to go for is. And let's say I'm only interested in a missense variant. So I'm going to say consequence is missense. And I'm going to click add. That should filter my table and I should be only left with the missense variants. Here we are. Just as I said, now we only shown missense variants. You can see their pathogenicity predictions and lots of other informations. You can also combine multiple filters, so web gives you quite a lot of flexibility. You can see it's lots of data with many links linking you out to different resources. If you wanted to download that data and work on it on your machine, you can do it here. You can see we support different download formats like VCF, web, and text format. You can also export this data to Biomart, which I'm going to be talking about in our last webinar in the series. Okay, great. That's all I wanted to show you when it comes to the web. Let me just quickly go back to my presentation to wrap up for today. We have another webinar this Thursday at 4 p.m. BST, British Summer Time, run by Astrid. Astrid will be talking about comparative genomics with Ensembl, and during this webinar, she'll take you through the gene trees and homologs, but also whole genome alignments between pairs and groups of species. If you have any more questions, please feel free to keep asking them. I'm going to keep the webinar interface open for a couple of more minutes. If you'd like to go through the questions or ask new ones, please feel free to do so. As I mentioned before, you can download the webinar materials from our online page. You can find both the slides and demonstration and the video itself under this link below. Furthermore, you can find the exercises and the solutions so that you can practice in your own time. If you need any help with the exercises, you can also use our living document. Please feel free to type any questions there. We'll keep checking them after the webinar. If you wanted to ask us questions privately, do send us an email at helpdesk@ensembl.org. Also, if you wanted to know more, we have more online courses, longer ones like this webinar series, but also shorter ones and animations. You can find them on our online channels. We also have public mailing lists that you could join. Finally, follow us on social media. We have a Facebook and Twitter account, but also Ensembl blog. To finish off, please do cite us if you're using our resources. Ensembl is free to use, but we do appreciate citations, which helps with our funding, but also keeps us informed on who's using us and how. Finally, I'd like to thank the entire Ensembl team of around 70 people. Here we have a photo from our last team retreat. And also, I'd like to thank our sponsors. Thank you everyone for joining today, and hopefully see you next time.